Good morning, students. Today we are going to study about monoclonal antibodies. Regarding the history of monoclonal antibodies, it was way back in 1975 when Kohler and Milstein they they were they prepared a method for preparing they devised a method for preparing monoclonal antibodies, and during this process, during the synthesis of monoclonal antibodies, they were able to show clonal selection theory, in which the technique used was that there was a fusion of normal B-type lymphocyte cells with malignant cells, that is myeloma cells, that is cancerous plasma cells. But the medium which was used for isolation of hybrid cells, called as hybridoma cells, was HAT medium. And in this HAT medium, it contained hypoxanthine, aminopterin and thymidine. This selection medium was used for two parent cell lines. As we go further, I will tell you. The principles. The hybridoma technology, it is a method by which we prepare number of identical antibodies called as monoclonal antibodies. When I say identical antibodies, I would like to emphasize that antigens, most of the antigens, they are having more than one epitopes. They are having multiple epitopes, and therefore they are inducing proliferation and differentiation of variety of B cell clones, each derived from a B cell that recognizes a particular epitope. So the resulting mixture, that is the resulting serum, is having heterogeneous mixture of antibodies, each specific for one epitope. Each antibody is specific for one epitope. So a polyclonal antibody response is being generated. A polyclonal antibody response is generally advantageous for organisms in vivo conditions because it increases the immune protection in vivo conditions, but it reduces the efficacy of an antiserum for various in vitro studies. So, the research was for developing monoclonal antibodies. Monoclonal antibodies, they are derived from a single clone and they are specific for a single epitope. Whereas polyclonal antibodies, they are having B clones which are recognizing more than one type of epitope. So it's a heterogeneous mixture of antibodies, each specific for one epitope in case of polyclonal antibodies. But monoclonal antibodies, they are specific for a single epitope and they are derived from a single clone. It was in 1975 that Kohler and Milstein, they devised the method for preparation of monoclonal antibodies, and in 1984 they got the Nobel Prize for that. They use the hybrid cells, that is a technology wherein the fusion of B lymphocytes with myeloma cells or tumor cells, or you can say these myeloma cells are cancerous plasma cells, they were fused. The cancerous myeloma cells, they have a property of replicating, that is they have immortal growth properties associated with them. And the antibodies, they are produced by the B cells. So, this way, the two cells, they are used for production of hybrid cells. The principle behind this was that a clone of B cell stimulated against a single epitope of antigen is fused with malignant antibody producing myeloma cells to produce a hybridoma cells. This hybridoma cells, as I told you, it has two properties associated with it. Firstly, it produces monoclonal antibody of same antigen specificity. Secondly, it multiplies indefinitely, producing a clone of identical cells. The resulting clones of hybridoma cells, which secrete large quantities of monoclonal antibody, they can be cultured indefinitely, and the technique for producing monoclonal antibody, it gives a powerful and a versatile tool to the immunologists. Now, when we talk about B cell hybridomas, they are produced by polyethylene glycol to fuse myeloma cells with B cells from animals that have been immunized with the antigen against which one wants to make the antibodies. The myeloma cells, they contribute immortal growth properties to the fused cells and the B cells, which are capable of limited growth in vitro. As all of us know that plasma cells, when they become cancerous, they multiply rapidly, and when they multiply, they do not have a lifespan. But if you talk of plasma cells, they multiply, they produce antibodies, and then they die out. So this is the difference between myeloma cells and the plasma cells. The myeloma cells, they contribute immortal growth properties to the fused cells, and the B cells, they are capable of limited growth. They contribute to the genetic information for synthesis of specific antibody of interest. What happens when there is a fusion between the B cells, that is your spleen cells, they are containing antibody producing B cells. With myeloma cells, polyethylene glycol is used, and not all the cells, they fuse, and they produce a complex mixture of unfused myeloma cells and unfused spleen cells, as well as number of fused cells. Among the fused cells, there are three different combinations which are being formed. The three different combinations, first is unwanted fusion of B cells with the B cells, or unwanted fusion of myeloma cells with the myeloma cells, and the third is a desired fusion of B cell with the myeloma cells. The condition of the procedures must selectively allow the survival and growth of B cell and myeloma hybridoma cells. These myeloma hybridoma cells, they are actually these mutants which are deficient in one of the nucleotide salvage pathways. That is, these myeloma cells, these myeloma hybridomas which fuse with the B cells, they are incapable of having one of the nucleotides for synthesis of DNA. So there are two pathways by which the cells can form the DNA. First is de novo pathway, another is salvage pathway. But these myeloma cells, they are deficient because of previously selected mutations for one of the nucleotide salvage pathways, which makes them not able to be grow in the hat medium. Now, what is this hat medium? Hat medium is the medium which contains hypoxanthine, aminopterin, and thymidine. This medium is used to kill unfused myeloma cells. If the mixture of hybridomas and the unfused parental cells is placed in this medium, the parental myeloma cells, they cannot survive. The B cells and the myeloma cells, they can survive on the hat medium, because the B cell is contributing the missing enzymes which are there for the salvage pathway. Although the unfused B cells, they are able to survive in the hat medium, but they do not survive for an extended period. Hat selection, it depends upon the fact that the mammalian cells, they synthesize nucleotides by two different pathways, as I told you, de novo pathway and salvage pathway. De novo pathway, in which the methyl or formyl group is transferred from an activated form of tetrahydrofolate. What happens is, when there is an active transfer of methyl or formyl group to activated form of tetrahydrofolate, which is blocked, this pathway is blocked by aminopterin, which is present in the hat medium. And when the de novo pathway is blocked, the cells utilize the salvage pathway. And when they have to use the salvage pathway, they overcome this blockage by converting purines and pyrimidines directly into nucleotides for synthesis of DNA and RNA. The enzymes that catalyze the salvage pathway, there are two enzymes. First is the HGPRT, that is hypoxanthine guanine phosphoribosyl transferase, and the other is thymidine kinase. A mutation in either of these two enzymes will block the ability to use the salvage pathway. Hat medium contains aminopterin, which blocks the de novo pathway, and it has hypoxanthine and thymidine to allow growth by the salvage pathway. Therefore, cells that are lacking the enzymes, that lack the either HGPRT or TAK enzyme, they will die in HAT medium because they lack the ability to use the salvage pathway. But in hybridoma technology, what happens is that the myeloma cells, they are usually double mutants. They lack the enzymes of HGPRT and therefore they are deselected in the HAT medium. So they have lost the ability to produce immunoglobulin G mutants. And the other fusion partner, that is the spleen cells, which are containing antigen activated HGPRT B cells, these cells, they have the capacity to utilize the salvage pathway for hypoxanthine to the hybridomas, thereby enabling their survival in the HAT medium. So the hybridoma cells, they will utilize the capacity of the B cells to multiply and to survive in the HAT medium. Once the antibody secreting hybridomas they are obtained, they will be screened for desired antigenic specificity. When they are screened for desired antibiotic specificity, the desired antibody, that is they will produce antibody which is specific for an antigen and it will be used for immunization. Others will be specific, but they are will be specific for unwanted antigens. And the screening technique which is used is radioimmunoassay technique or ELISA technique. What happens in radioimmunoassay techniques, there is generally a competition between binding of radio-labeled antigen and unlabeled antigen to a high affinity antibody. So, antigen which is mixed with antibody at a concentrate, it saturates the antigen binding sites of the antibody molecule, and increasing amount of the test sample containing unlabeled antigen of unknown concentrations are added. Since the antibody does not distinguish between labeled and unlabeled antigen, so the two kinds of antigen compete for the available binding sites on the antibody. With increasing concentrations of unlabeled antigens, more labeled antigens will be displayed from the binding sites. This ratio of antibody to antigen is chosen such that the mixed number of epitopes presented by the labeled antigens always exceeds the total number of antibody binding sites. This ensures that any unlabeled antigen added to the mixture will compete with the radio-labeled antigen for the limited supply of antibody, and a small amount of unlabeled antigen added to the assay mixture of the labeled antigen antibody will cause a decrease in the amount of radioactive antigen bound. And this decrease will be proportional to the amount of unlabeled antigen added. So, a standard curve can be generated wherein the unlabeled antigen samples of known concentrations, and from this plot the amount of antigen in the test mixture can be precisely determined. This is one of the method. Another is ELISA method, which you can study later on. Now, when we talk about identification hybridomas of the desired specificity, they are being recloned and the culture is truly monoclonal and then propagated in one of the several ways to produce the desired monoclonal antibody. Next time we will talk about how the screening of hybridomas is being carried out, that is antibody secreting hybridomas, which once obtained they must be screened for desired antigenic specificity. Depending upon the method, either it can be a radioimmunoassay method or ELISA enzyme-linked immunosorbent assay method. Thank you, students.